Okay, let's have a class. First of all, we want to go over some of the results from last week. And I don't think you have any problem about APC. This is APC. You have a red pink dots on the petrofilm, that is APC. So you can go ahead to count how many are there. They multiply by the dilution factor. And you have two petrofilm, one is minus 10 to the second, another one is 10 to the fourth. So obviously 10 to the fourth should be countable. And 10 to the second might be a lot. Okay? That is easy, not a problem. But what we are much more interested in is this coliform and the E. coli. This is from one of the students, look at the very good. Majority of them are pink, and the red, that's coliform. And you see a red, did you see a blue colony there? And also there's a gas bubble, that is E. coli, which is generic E. coli. That means this sample is fecal contaminated. And I also wanted to say, do you know our drinking water system? There is a standards for coliform and fecal coliform. What is that? Is zero tolerance. That's why in the United States, your tap water ideally is drinkable. Now, of course, there is another issue regarding the soft water and the hard water. That's the calcium and the magnesium, the concentration. That's another topic. But from microbiology standpoint, in the United States, it is drinkable because the drinking water based on the EPA regulation, our drinking water, we also say municipal tap water, which is required zero tolerance for generic E. coli. The reason is if you have one cell of generic E. coli there, which means it's fecal contamination. So that's important. We need to keep in mind, although this test is easy, but it's very meaningful. And you see here, obviously there is one E. coli there. It's gas bubble and the blue colony. So remember, what does E. coli looks like? It is blue colony and with a very obvious, you see a gas bubble. And this is generic E. coli. And if you see a pink colony, that is coliform. Now what is the total coliform? They add up together is total coliform. And you can see even our WVU pound water, the coliform actually is pretty huge. Let's say this is 10 to the second, 100, you see at least this is like a 15, 20 cells there. That means there's 100 generic E. coli and pretty much 3,000 coliform there. So that's important to tell you something. Okay, they did a good job. Very good job. Does anybody else find the generic E. coli? Which means you plates, you petrofilm, the pink one, you see there is a blue colony there. Now, for example, this student doesn't have anything. It's just a coliform, it's not E. coli. Does anybody else who have it? Okay, that's good. We at least have one sample in the whole classroom, which looks really nice. Okay, very good. Okay, today's work is simple. You may go home 10 minutes, but we will do it because we talk about the antibiotics, talk about could be blood test, but we haven't done yet. So we're going to do is could be blood antibiotic resistant test. Okay, first of all, we will be very briefly review what is called antibiotics. What is antibiotics? This is a metabolic chemical, metabolism chemical, generated from one microorganism used to kill or inhibit another microorganism. And I said this first microorganism most of the time is fungi. And it is could be yeast or could be molds sometimes. Let's say penicillin. And this another microorganism, most of the time we mean it's a bacteria. And what means a kill? That means this terminology called cyto, and what means inhibit is static. Okay, so that's very briefly review what we have. The second one, we had a Kirby-Boer test. So how to do the Kirby-Boer test? Then that's some procedure we need to know. First of all, you and your bench partner, we will do 2 plates for everybody. One plate will do E. coli, another plate will do lactobacillus, because we are representing each of them is gram-positive and gram-negative bacteria. So we will choose gram-positive bacteria is lactobacillus. And we will choose gram-negative bacteria, which is E. coli. So how are we going to do it? First of all, you will prepare is to 0.1% nutrient broth. This is 0.1% nutrient broth. And you will using is nutrient broth like this. And then use your loop to pick the colony from the slants. And then put inside. And you shake very well. You will see the turbidity there. Now, originally, we should have a standards for that. So, let's say the slants right here. You use loop, pick them, and you go here, and you're gonna be mixing. Okay, same thing, you mix here. Let's say this is E. coli. This is lactobacillus. Now, how turbidity it is? Originally, ideally, there is a standards, because otherwise you're gonna put a different type of the bacteria or different concentration of the bacteria. They have to meet a standards called 0.5 McFarland standard. What it means 0.5 McFarland standards, approximately 10 to the 8 CFU per ml, or we call cells per ml. This is the first thing we need to do. So, ideally, once you have the solution prepared, we will prepare is a silicone made 0.5% McFarland standards, but I don't have it right now. Then we will compare these two turbidity. Once they are matched with each other, which means your concentration goes there, is 0.5 McFarland standards, indicate approximately 10 to the 8 cells per ml. But today, you don't have to do that, because we know if you pick about 1 or 2 colony from these slants, you put in the 0.1% nutrient broth, it is approximately 10 to the 8 cells per ml. Okay, we did a couple of years already. So that's the first thing. Then the second thing, we need to add agar plates. This is the agar plate is a little bit different. This is not a traditional nutrient bloss, but looks like very similar. This is actually Molo Halton agar. I need to make sure that spelling is correct. So let's see what we have. The agar's name, I used to just call it MH agar, but this has to be Molo Halton agar. So M-U-E-L-L-E-R, Molo Halton agar. This is a very special agar for antibiotic resistance. Okay, once you have this agar ready, what you do? The same thing at the back you have to label. So label your information at the back. Then what you do? Using cotton swab right here. The cotton swab is right here. The same you do soil culture. You add onto the, dip into the solution. And then took it out. Averagely spread on the surface. So how we do it? You will have to do a very gentle perpendicular streak using cotton swab. Okay, sterilize the cotton swab. So we're very gentle like this. So that is sterilized cotton swab. Then you have to turn 90 degree. Remember, do not put the cotton swab back to the solution because you'll carry more bacteria. Turn 90 degree, do a horizontal one. Okay, so do it again. Very gentle, like this. Okay, now what we do? We need to use a cubby-bore dispenser. $2,000, very expensive, I tell you, although it's always broken. Cubby-bore dispenser. What we need to add in it? We need to add these type of the very tiny disc. Every disc which is containing antibiotics. And the concentration is already there, and it is already being fixed and pre-made. So we need to add them in and then press the button and do the dispenser. So what we're gonna do today, what we're gonna add them to them today, we need to add tetracycline. Okay, tetracycline is TE. That's an acronym for tetracycline. And the concentration what we're gonna use in here is 30 microgram. That's tetracycline, the first one. A different lab, I have to use a different one because I don't have that much enough for every student. Second one, nalidic acid. Nalidic acid's acronym, I will just say Na. And the concentration here is also 30 microgram. Now remember, antibiotics, their concentration is limited to microgram because you have to do the smaller the better. Another one, what we're going to do is kanamycin. So right here. Kanamycin, the acronym you can write is Ka. And that's also 30 microgram. Okay, after we using dispenser, we add a disc onto the plates. We will do 35 degrees Celsius, 24-hour incubation. Now Thursday we don't have the lab, but you can come back to see your plates. I'll have the ruler or on the bench, you can grab it to measure. Because if you do the observation next week Tuesday, it will be horrible sometimes. So I encourage you after exam, you can come back to see it. The first lab, I know you're still preparing for those cycles and extra points. After exam, you can come back to lab. So ring open the lab like 12 o'clock, put all the plates out, and they can grab and to measure. So what are you going to see it? You will see more or less there is a transparent zone. Okay, something like this. Something maybe a little bit small. And there could be something huge. That's transparent zone, which is inhibitory zone. Now what are we going to measure? We will measure is the diameter from here. And also the diameter from here. And the diameter from here. Okay, so we're going to measure diameter. Of inhibitory zone. And then you record it. Okay, then you record it. Okay, here is the key. Every inhibitable zone is representing the bacteria response to the antibiotics. So I'm going to just give you one example. Let's say you have a tetracycline, which is 30 microgram. The bacteria response to the antibiotics basically have three, which is S, susceptible. We don't say sensitive for antibiotics, because you know, some of you is allergic to penicillin. We say we are sensitive to penicillin. Let's say Madeline cannot give muscle injection of penicillin. So we say Madeline is sensitive to penicillin. Because when we do the penicillin, we have to do the skin test. If you have a swelling comes out after 24 hours, which means you are allergic and sensitive to penicillin. So at that moment, we use the words is sensitive. Here, for bacteria to the antibiotics, we call it susceptible. Okay? We don't say sensitive. Susceptible. Okay, now we know if some of them are susceptible, of course we have resistant. Which means you don't have much effect. Now between that, intermediate. So let's compare the table. And on your fact sheets, at your fact sheets, the last page, we had a table for that actually. So we just give an example, tetracycline. Tetracycline we see is more than 19. Okay, it is 15 to 18. And the last number is actually 14. And this is all millimeter, mm. So what this means? On Thursday, you're going to measure the inhibitory zone. Just record on your notebook. Then next week, Tuesday, we could talk about that. If you see your tetracycline, this diameter is 20 millimeter, let's say, then we will say the bacteria you added here, either E. coli or lactobacillus, is, sorry, the other one. Sorry, I made a mistake. This is more than 19 millimeter. And this is 14 millimeter. If you see your inhibitory zone, let's say it's 20 millimeter, okay? Then what this means? The bacteria you put there, E. coli or lactobacillus, is susceptible to tetracycline. Let's say some other people find your lactobacillus, the inhibitory zone is only 12 millimeter. So what we say? Although it still inhibits bacterial growth, but compared to this table, we will say your lactobacillus is resistant to tetracycline. So clinically, which means for this patient, you cannot use tetracycline to do the treatments because the bacteria cause resistance. Now somehow, if you get like 17 millimeter, that means it's intermediate and clinically you still cannot use it. Okay, so that's what we do. I have a different antibiotics right here compared to this table. Not everything is covered, but I will have a full table for you next week, Tuesday. So what are we going to do today? We're going to prepare the Kirby boil test. We're going to do the incubation. And then on Thursday, you come back to measure it. I'll have the ruler put on the bench, you just measure. The ruler is just a general ruler. Okay, regular ruler like you'd use for the math class. And then, next week, Tuesday, we'll talk about the results. Okay? So go ahead to do it.